Thank you all for tuning into my presentation on the genomic landscape of Ornithobacterium rhinotracheale in commercial turkey production in the United States. Ornithobacterium rhinotracheale, or ORT, is a bacterial respiratory pathogen that can infect a variety of avian hosts, but is particularly severe in turkeys. ORT causes respiratory distress, including coughing and sneezing, as well as reductions in feed and water intake and increased mortality. Post-mortem, gelatinous lungs are sometimes seen, or lung consolidation with a fibrinous exudate, as well as air saculitis. Co-infections of ORT with other pathogens are common, and often result in higher condemnation rates than either pathogen alone. E. coli is one of the most common sources of a co-infection with ORT, but avian influenza, Newcastle, infectious bronchitis virus, and avian metapneumovirus have also been reported. ORT was recently ranked as one of the top 10 current health issues facing the turkey industry, falling in third behind colobacillosis. This is presumably due to the significant economic losses associated with ORT, as well as concerns for animal welfare. ORT is sometimes treated with antibiotics, the most common of which are tetracyclines and erythromycin. These charts to the left were taken from a recent report of on-farm antimicrobial use in the United States. You can see that for all of the turkeys that received tetracycline in 2017, 5% was specifically for ORT. For all of the turkeys that received erythromycin in 2017, 40% was specifically for ORT. Other antibiotics used include penicillins, sulfonamides, and lincomycin. However, antibiotics aren't always effective due to antimicrobial resistance. Some studies have attempted to describe the antimicrobial resistance of ORT isolates. However, there are no standard MIC breakpoints for antimicrobial resistance of ORT, limiting the objective classification of isolates as either susceptible or resistant. Conversations with poultry veterinarians have indicated an increasing trend in resistance to tetracycline drugs, but this is yet to be measured. The lack of information available on antimicrobial resistance of ORT, as well as the industry-wide shift towards antibiotic-free production, really emphasizes the need for preventative measures for ORT. However, there is no commercially available live vaccine for ORT in the United States. Therefore, producers must rely on autogenous vaccines produced from flock-specific bacterial cultures for ORT prevention. But there are very few studies on the efficacy of autogenous vaccines in commercial birds. Similar to the concept of autogenous vaccination is controlled exposure to ORT, with the goal of conferring protection to ORT by introducing the bacteria to young birds. Again, there is limited information available on the successes or failures of this method other than anecdotal reports. One potential barrier to the development of a successful ORT vaccine could be the large knowledge gap in ORT genomics. Very few studies have looked at ORT at the whole genome level. Most studies have used multi-locus sequence typing to categorize isolates, but this method is based on the allelic profiles of 7 genes and therefore doesn't capture genetic differences that may be outside of these regions. The purpose of this study is to better understand the genomic landscape of ORT in commercial turkey production in the US, which may ultimately provide insights into the selection of the most appropriate autogenous vaccine strains. This was a retrospective, cross-sectional study using ORT isolates collected from commercial turkey operations. 4 major U.S. turkey producers participated in this study, as well as 2 independent turkey growers. Isolates were recovered from clinical swabs collected from respiratory lesions of commercial turkeys displaying typical signs of ORT as identified by field veterinarians. Swabs were streaked onto sheep's blood agar and incubated microaerophilically for 48 hours at 37 degrees Celsius. PCR was used to confirm the isolation of ORT, then whole genome sequencing was performed at the University of Minnesota. Now I'll briefly walk you through the bioinformatic pipeline used to analyze the whole genome sequences. First of all, genomes were assembled and annotated. Multi-locus sequence typing was performed. Short nucleotide polymorphisms, or SNPs, were identified in order to highlight regions of genetic variation between strains. Then these SNPs were used to build a phylogenetic tree. A pangenome analysis was conducted, and the pangenome represents all of the genes present in all of the ORT genomes. And this was used to identify differences in gene content between isolates and different phylogenetic clades. Lastly, mobile genetic elements were identified and annotated. This study included a total of 157 clinical isolates collected from 1999 to 2019. The different producers are designated as companies A, B, C, D, or an independent grower. There are 27 isolates from Company A, 53 from Company B, 34 from C, 41 from D, and 2 from the independent growers. This is the resulting SNP-based phylogenetic tree. The different colors represent the different turkey producers. A, B, C, D are independent, and isolates are labeled by collection year if that information was available. You can see that all of the clinical isolates fall into 4 distinct phylogenetic clades, and these clades are labeled in the outer ring. All isolates within a single clade belong to the same sequence type. All of the isolates from clades 1 and 2 belong to sequence type 1. Isolates from clade 3 belong to ST34, and isolates from clade 4 all belong to ST35. Notably, all companies had clinical isolates collected in the same year fall into multiple phylogenetic clades. This indicates that multiple ORT strains are likely circulating at the same time instead of a single dominant strain. Interestingly, Clade 1 includes isolates collected from 1999 to 2018. It also includes the reference strain from 1995, demonstrating that this particular strain of ORT has persisted in commercial turkey operations for over 20 years. Company A, depicted in purple, had the majority of clinical isolates fall into Clade 1. This clade also contains a 2015 vaccine strain from Company A. The fact that the majority of clinical isolates from this company fall into Clade 1 may be indicative of vaccine failure, as the vaccine strain matches the pathogenic strains that were circulating at the time, but was not successful in preventing disease. Company C, shown in green, had isolates fall into 3 phylogenetic clades. The 2017 vaccine strain from Company C is in Clade 2, which also contains a few other clinical isolates from 2017. However, the majority of 2018 and 2019 isolates from this company fell into Clade 4. And in 2019, Company C switched vaccine strains to a strain from Clade 4. Clade 4 also contains 2 isolates, both from Company D, that contain a specific insertion sequence with antimicrobial resistance and virulence genes. This is an annotated map of the insertion sequence, with the different colors corresponding to different types of genes. Notably, the sequence contains genes conferring resistance to aminoglycosides, chloramphenicol, erythromycin, and beta-lactam antibiotics. This sequence also includes genes, shown in orange, associated with bacterial virulence or fitness. It's possible that the ORT strains containing this insertion sequence may be more virulent than the other ORT strains that lack this particular sequence. Another mobile genetic element was identified, plasmid POR1, the one known plasmid associated with ORT. This plasmid was found in all isolates from Clade 4, and was not found in any isolates outside of Clade 4, indicating that this plasmid may be clade-specific. Here is an annotated map of the POR1 plasmid. This plasmid contains genes associated with resistance to heavy metals, shown in purple. Note that these metals include copper, which may be of concern since copper sulfate is sometimes used as a disinfectant in water lines. POR1 also includes virulence-associated protein D, shown in orange. It is possible that isolates from Clade 4 may have an enhanced virulence potential due to the presence of this plasmid, and in particular, the 2 isolates that contain both the plasmid and the insertion sequence. This was the first large-scale study to perform comparative genomics of clinical ORT isolates. All ORT isolates fell into 4 distinct phylogenetic clades. This could eventually lead to the development of a clade-specific PCR, which could provide important information on ORT isolates to veterinarians when whole genome sequencing isn't readily available or financially feasible. Overall, the same strains of ORT have persisted in commercial turkey operations across the United States for nearly 20 years. This may be partially due to the vertical integration of the poultry industry, allowing for transmission of the same ORT strains between different stages of production. This could also be due to the failure of autogenous vaccines to confer protection against ORT strains that are genetically similar or genetically dissimilar to the strain used in the vaccine. Also, there are strain-to-strain differences in gene content which may result in differences at the clinical level, but this needs further investigation. The identification of genes associated with antimicrobial resistance or resistance to heavy metals further emphasizes the need for ORT prevention, ideally through more effective autogenous vaccines or controlled exposure methods. This study does have a few limitations, however. The first being that this was a cross-sectional study, so we can't determine a causal relationship between the vaccine strains used and the clinical isolates that were submitted. Another limitation is that we did not have phenotypic data available to confirm whether the antimicrobial resistance genotype would actually result in a resistant phenotype. However, the absence of MIC breakpoints for determining susceptibility or resistance of ORT would have prevented the objective classification had this been attempted. But this does raise the question of how veterinarians can use previous SIR data to inform the development of ORT breakpoints in the future. That concludes my presentation, but I would like to acknowledge my funding sources, as well as the laboratory staff that contributed to this study. My email is there at the bottom. Feel free to email me with any questions you might have. Thanks for listening.